the difficulty of isolating and culturing many marine microorganisms are regarded as viable but non-culturable. So, which one of the following is the suitable method to hunt them? So, first of all, it is saying about the in situ cultivation. In situ cultivation is the age-old cultivation. Before the coming of the synthetic or minimal medias, this kind of cultivation techniques are there. So you are cultivating in the open air, in the open environment. How will you isolate them? It will be very much difficult. And the question is saying to remove the difficulty. So there some contaminations will be there. Will never be able to identify the actual one. So it is not at all feasible. And here your bacteria is VBNC, means it is non-culturable, but it is showing its activity. You are not able to see them, you are not able to grow them, but you can identify about their functions. They are showing some functions, but they are not growing. So what might be the reasons behind this? So it could be they are not getting adequate kind of nutritions in the culture media, or might be they need certain kind of specific nutrients, like certain rate limiting nutrients are there, certain specific nutrients you should provide. Might be their salinity or the oxygen concentrations that you have to modulate, and also it could be dependent on the metabolites that is producing from another bacteria or another organism. So in a monoculture, in a pure culture, when you want to just only culture a single type of bacteria, it is not possible to grow them. So that might be the reason. For that, you are not even able to grow them in the laboratory condition. So now you are not able to grow them. At least a plate you are not able to grow, and you are not able to grow them, and you are thinking to grow them in a large bioreactor. That is very much difficult. Even for the conventional microorganisms that you can grow in the lab, so doing their upscaling to grow them in a bioreactor itself is a difficult thing. So like different kind of parameters are there, and you have to optimize them all, like the nutrient, the substrate. You have to, that substrate concentration should be equal, should stay same from the up to the bottom of the bioreactor. Maintaining this kind of things, that shear stress and other kind of things, like you have to provide the specific amount of oxygen, so that oxygenic or anoxygenic conditions, that means the aerobic or anaerobic conditions should be maintained properly, the pH, temperature, maintaining everything, that is very much difficult. Mm, and so this growing them in a large bioreactor is not at all possible. So the next option is the barcode sequencing. The barcode sequencing might be an option if we are having the sequence ready in the barcode dictionary. So barcoding is the, like, it is a specific sequence that is present in a specific species or a specific microorganism. And after isolating the microorganism, you will get the DNA. You will extract the DNA sequence, and thereafter you will identify that constant region, and thereafter you will go to find the similarity into the dictionary. If you are getting an exact match in the dictionary, that means you can identify which organism is it. So you're not able even to culture, and so you are not having any kind of information about its barcode. So barcode sequencing is not even possible. So the only way is the metagenomic analysis. So how to do this kind of analysis? You will extract the environmental sample. So from environment, you will get the eDNA, the environmental DNA. So you will extract the DNA. Like as an example for this, you are you took the water from the sea level, from a marine resource, and you have filtered the water. Now you have the cell debris. From there, you will extract the eDNA. And like just think, so it is an environmental sample. So lot of type of bacteria will be there. Lot of will be there. So if you want to isolate this D, you will get every kind of bacteria. So at first, it is not able to identify which one is D, which one is E, which bacteria comes from where. It is very much difficult to identify. So here, at least that means you are having a pool of genes of DNA. So from each bacteria, you will get a bacterial genome will be very much long, and it is also not very much feasible to sequence them on a one shot. So now you have to share them. The sharing will be in a non-controlled process. So the sharing can happen at any position. So anything will be possible. So thereafter you will attach. So now that is those are the short DNA sequences. So that is the whole genome, and you have divided them into short sequences in a non-controlled manner. So the sequence, that short sequence could be anything. You don't know where the cut was there, anything, because you are using the physical methods to to break the long DNA in a short DNA sequences. Now you will attach some adapters. So adapters are the known sequence of DNA, and thereafter we will go for next generation sequencing, or before that you can go for PCR to amplify their number. And now you will go the next generation sequencing, and here you will get the DNA sequences. You now, till now, you have made from this long genome, you have made certain short DNA molecules, short length of DNA you have prepared. And so these DNA sequences were unknown to you. Now after running the NGS, you know the sequence. So all the sequences are known to you. And now depending on that, you will make a contig. So how to form this contig? So I told you the sharing will be in an uncontrolled pattern. So the sequence can happen anything, like for one short sequence and another short sequence might have a, that is, complementary sequence region. So if the sequence are getting similar, that means, so that means the first sequence was this, and thereafter till does this, and thereafter, so this is the same sequence, and thereafter it will be this, and here also this is, and thereafter it will be that. So when you will make the total, you will annotate it in a total form. This will come like, so the blue is here, it will come like this, and thus you will synthesize the whole genome. Now you know the whole sequence. This kind of sequencing is known as the metagenome assembled genome, that you will tell as the MAG. You have assembled in a metagenomic order, and there it could be from A, B, C, D, E. For each bacteria, you will get this each kind of MAG. For each bacteria, you will get at least, you will get one MAG, and this MAG will be stored in a bin file. So for different MAG, you will get different bin files, and each MAG will contain the sequence of a single bacteria, single microorganism. So that was the conformational part. Now, if you, that was the conformation based metagenomics. Now, if you just want to know that was the mag. You now know the whole genome sequence. Just you want to know the taxonomy, or just you want to identify this bacteria whether that was previously known or not. Then you can go for the OTU. So here you just have to have the 16S RNA sequence, and this RNA sequence is having different regions, like that is the variable and constant region. So depending on this sequence, you can determine the OTU. So depending on the sequence similarity, you will get the OTU formation. This OTU will help you to make the operational taxonomic units. So now you know whether that bacteria is present in which taxonomic unit. That is the OTU. And another thing, now you know every kind of thing like their genetic material and their taxonomic situation, taxonomic establishment, but still you want to know the functionality of that bacteria, of that organism. So what to do then? That is the function-based metagenomics. Now you will again be having short DNA fragments, and here you will attach it with the expression vector. Um, all the you will all the by using all the fragments you will make a expression vectors, and thereafter you will transform them into bacteria. Now you are having a like a library of bacterial expression vector that will produce your target proteins, and depending on that you can determine the function. Now you will know the function of this DNA sequence. Thus you will come to know what kind of function this your target bacteria is producing. So thus it gives the so much power to the metagenomic analysis to determine the VBNC organisms. So let's go to the next question. You can see two statements on McConkey agar. So McConkey agar is very much popular in bacteriological labs, and it is both, means it is the differential media, differential, as well as selective media. Before going to that, this kind of differentiation, so we should know the content of this agar. So that is the one is the that is written over here, bile salt and crystal violet. That is CV, and bile salt, and third one is the lactose. Lactose is the carbon source, and fourth one is the that is the pH indicator. Here it is neutral red. So you can see the negative control plate. So it contains this kind of nutrients or reagents. So it is saying that McConkey agar is a media used for recovery of gram-positive bacteria. It should be gram-negative bacteria, not the gram-positive bacteria. So here comes the term differential and selective. So it is like selective. Selective means it is not allowing some bacteria to grow. Like only the gram-negative bacteria they can grow in this kind of plate, and if this kind of nutrients are providing in the culture media, only the gram-negative bacteria can grow, and the gram-positive bacteria will not be able to survive under this condition. That makes it selective. So in the culture media, we give bile salt and CV. So both are responsible for destroying the gram-positive bacteria, but not the gram-negative bacteria. Thus this agar is a selective agar. And what makes it differential? That is the presence of lactose. So two kind of gram-negative bacteria, they can differentiate between two kind of gram-negative bacteria. That is, one is the lactose fermenting bacteria and another one is the lactose non-fermenting bacteria. So in this picture you can see this is the lactose fermenting bacteria and this is non-lactose fermenting bacteria. So equally is a like lactose fermenting bacteria. And if the lactose is the carbon source, so the E. coli or other kind of lactose fermenting bacteria will utilize this resource and thus they will decrease the pH. They are using it, they are using lactose as a metabolite and they will produce certain kind of chemicals that will be acidic in nature and thus the pH of the media will decrease down and there is the indicator neutral red which will make this kind of color. This will change in color and thus you will be able to determine there is a lactose fermenting bacteria. And the another part, when the color was not changed, although both are negative bacteria, gram negative bacteria, but this one is lactose fermenting and this one is non-lactose fermenting. So option 2 is saying the bile salts and crystal violet in McConkie agar inhibit the growth of some bacteria. Yes, it inhibits the gram positive bacteria. So the fourth statement is right. 4 different unidentified bacteria are there, ABCD, and they are having the thymine in a 38%, 26%, 24%, and 12%. So if we take the T in the genome, the amount of T will be equal to A. The thymine and adenine amount should be same and the G and C amount should be same. So here, so the if we take A, B, C and D, T, A, G and C, so the percentage of A 38, 26, 24 and 12. So the percentage of G plus C will be here, it will be 62, here it will be 74, 76 and 88. So now one of these 4 species was isolated from a hot spring. So they are having the 64 degrees centigrade. We all have the knowledge that between T and A double bond is there and G and C triple bond is there. This is a very simple question and it is staying on the 64 degrees centigrade hot spring. That means it is a extremophilic bacteria. So if the percentage of T and A is higher in this bacteria, then the bond will easily be broken away. So the DNA would not be able to stay in its conformation, in its double strand conformation. So the percentage of G and C should be higher to maintain the conformation so that the bacteria can sustain at that temperature. The DNA should not be denatured. So that is a evolutionary selection. And here the D is showing the high percentage of G and C. So the option would be the fourth one. This question is about the gram staining, where in the assertion A and reason R, you can study the statements and there is a name we are getting, that is a mycoplasma. Mycoplasma is a specific bacteria that is not having a cell wall at all. So first we should have some knowledge on the gram staining and what is the basic principle of it. So the gram staining that differentiate the bacteria depending on their cell wall structure. cell wall conformation. So here you can see in the gram negative and gram positive case, for the gram positive, the cell wall is made by peptidoglycan. So that peptidoglycan layer is so much thick and it is exposed directly, it is exposed to the outside of the cellular structure. But here in the gram negative, that cell wall is also made by peptidoglycan, but that is very much tiny, that is a very much lean layer, and thereafter you can get a outer membrane. So gram negative cell wall is made by a thin peptidoglycan layer and it is not exposed to the outside. Now, when you are doing the gram staining, if you use after the fixation, if you use the primary stain, that is the crystal violet, then that will that crystal violet, this this stain is having a positive charge. So when you are using the crystal violet, it will directly come and bind to the negatively charged peptidoglycan layer. The peptidoglycan for gram positive is so much thick so that it can come and directly bind to it. And whereas for the gram negative, it takes time to enter through the that outer membrane. And the layer is very much small, very much thin. That's why it is not able to retract the color. Now, after the crystal violet treatment, we are using the iodine treatment. That is a mordant. So what is its work? It will fix the color so that after using the decolorizing agent, so that it can't go away. So to fix the color, like here the crystal violet is here present and it will stain the layers. And now we will use the mordant and the color will be fixed. But here there is the binding of the crystal violet and this peptidoglycan layer was not happening, or else if it was happening, that was very much small in number. The interaction was not that much strong. That's why even after using the mordant, that can't be fixed in the case of gram-negative bacteria. Now we are using the decolorizing agent, and thus the treatment of the decolorizing agent in both the cases. So here the color is fixed, it can't go away. Even after using the decolorizing agent, the stain will remain in the peptidoglycan layer. But here the interaction was not that much strong. That's why even after using the mordant, using the decolorizing agent will remove the stain from the layer. So the stain would not be there. And now we are using the another stain, that is the safranin. That is the safranin. That is also having a positive in charge, and it will come and slowly go inside and will bind here, will attach here slowly. It will take some time. And now we are not using any decolorizing agent, so it will not be removed. It will not going to be washed away. If even after that, if we use the decolorizing agent, then this stain will again be removed. So in the gram negative case, we will get the color of the safranin, and in the gram positive case, we will get the color of the primary stain, that is the crystal violet. Now that is the principle of gram staining. If certain bacteria that is not having the cell wall structure will not be even able to colorize it via the staining procedures. So mycoplasma is a bacteria without cell wall, that is right, and it stains negative in gram staining. So it doesn't at all stain in the gram staining. So that should not be right. But only the reason it is saying only bacteria that have a cell wall would stain positive in gram staining. So that should not stain positive in all the cases. It could be negative or positive. So the reason R is totally false. So if the R is false, then you have to say that A is true. R is totally false, but A is partially true. Even it should be, it is, it is stains negative in Gram staining. It should not be stained, but if there is no kind of option, then you have to write it that A is true. The partially it was true, but that portion is not actually perfectly true. But if you are staining, it can't even, it never can be in a positive way. But if there is no chance to be negative also, but still the answer is like that. So it should be, it should be not even negative, not even positive. But depending on the answer, we are taking the third one. Or we have reached to our last question for today's session, and there are some features, and you have to say which one is not contributing to the host tropism in the bacterial pathogenesis case. So, first of all, what is a tropism? So, like we know, every kind of pathogenic organisms has a specific kind of restrictions, a limitations, a host selection is there. Like the TMV, tobacco mosaic virus, can't infect a bovine or a human. Like same, the tuberculosis can't happen in the case of a plant. So every kind of bacteria or the pathogenic organisms, they are having a certain kind of restrictions. Like that is the host selection. And here it is saying the tropism. So inside the host, there will again be a selection. Like a HIV virus, it will stay, its tropism area is the TH cell, CD4 plus TH cell. So it will go and infect in that kind of cells. And for the tuberculosis, that bacteria will come and stay in the lungs. Its primary infection site is the lungs. Thus, if you know the tropism of your target bacteria, then you will be able to know it is present in which organ or which place in a body. That is called the tropism. So, till now, whatever understanding you have, I guess you can say the genomic information of pathogen and host both are important in that kind of case. Like in the previous video, I also have told about the CD4 plus cells that can have a, that could be a receptor for the GP120 protein, the glycoprotein 120 that is present on the surface of the HIV virus. So CD4 plus cells means the TH cells on the surface. This CD4 plus is present. That works as the receptor for GP120 protein that is present in the on the HIV virus. So both the genetic conformation for the host, here this is the host and this is the pathogen. So both the genetic information is very much important. If it doesn't have that CD4 plus, then it would not be able to infect it. And the same way happen on the inverse case. So both are important. Like, and the fourth option is saying about the adhesins and pilins. So both are kind of proteins. The pili protein you can see here. So both type of proteins are present on the surface of pathogen. And do you know a? Can you give me an example of a adhesin protein? That GP120 is a type of adhesin protein. So we also know that this kind of proteins are very much important to have a binding, to go and bind to the receptor site. So adhesin proteins are too much important. And now the pilins. So pilins protein are very much interesting thing. So they can form the sex pillets, and via this they can make a horizontal mode of DNA transmission. Horizontal DNA transmission. So from the pili, by making the sex pili, it can form a conjugation with another bacteria, and in that mode, that bacteria, if that bacteria is the pathogenic one, then it can also contribute, it can also give its pathogenic, that genetic material, to this non-pathogenic bacteria. Thus this bacteria could be a pathogenic one. Actually that is the evolution, how the bacteria was bacteria transform into a pathogenic one. That is also a horizontal gene transfer. This is also a technology, that is also a procedure. Thus the bacteria, that like of the pathogenesis were spreaded via the HCT procedures. So the pillins is a major culprit in that case, and also it is present on the surface. So those are the pillins. So it can go and bind to the surface of the host, and they can form a specific kind of, that specific kind of pillins only. There are also types of pillins like 1, 2, 3, 4. So specific kind of pillins can go and bind on the surface of the host, and it can form a twitching like of movement. So that movement will lead the adhesion protein towards the receptor, target receptor. Thus, it gives a very much crucial activity in the bacterial pathogenesis, and also it can make the biofilms. So from the pillin protein, it helps the cell to rapidly divide, and also to adhere and to go till the, that is the receptor of the cell. Thus the pillin makes a very important activity in the case of pathogenesis. So that is also important, that is also included in the bacterial tropism, in the bacterial pathogenesis. Now only the excluding part is the body temperature of the host. So thank you to stay with us till now, and if you are having any kind of difficulty or any kind of suggestions, please let us know in the comment box. And goodbye for of now, and we'll meet in the next video. Thank you.